David Liu and his team did it again. Just after we told you about how the prime editing recombinase duo can insert large DNA sequences, they just went and made it better.
In fact, if you remember in our Making the Cut series, we told you how it's possible to combine prime editing and recombinase to integrate large cargos.
As a quick reminder, prime editing is used to insert target sites that then the recombinase uses to do its magic and insert a large DNA sequence.
However, this elegant approach still suffers from some limitation, or that's what we thought until now.
You see, David Liu and his team realized that the main issue was that the recombinase they used, called BXP1, was not efficient enough. In fact, when they tried it on cells, where all the cells had the recombinase target sites already in their DNA, only 10 to 20% of the cells showed recombination happening.
And so they thought, if the protein we are currently using is not good enough, let's make a new one ourselves. In fact, what we didn't tell you is that David Liu and his team developed also a system called PACE, which allows to simulate years of evolution in a matter of few weeks.
As you know, evolution can take millions of years, and nobody's got time for that. So the PACE system simulates quick evolution by forcing viruses called phages through a very high selective pressure. This way, only those phages with very highly active versions of the protein of interest, in this case BXP1, are able to replicate.
And this process allowed them to isolate up to 40 different variants, almost all of them working better than the previous BXP1 recombinase.
So then they went and looked at what happened actually in these variants, and they found that the mutations that increased BXP1 efficiency were concentrated in two domains called NTD and CTD.
If one mutation was working, let's try to combine more than one, and actually they identified a magic trio which could work even better than a single mutation.
This new recombinase that they created was called Enhanced Evolve BXP1, because why not make names even more complicated? But for friends, we will just call the new editing system eePASSIGE.
And the eePASSIGE was put up to test against the original PASSIGE, the previous versions with only one mutation, and even its close competitor, which is called PASTE, and it could support all of them.
Indeed, they achieved over 3-fold increase in integration efficiency, and yes, that is a big deal, from under 10% to even up to 45%.
They even tested the system at different genes in different cell types and also with different cargo sizes.
Now you will be thinking that David Liu and his team will finally be able to take a break and enjoy the summer holiday. We're sorry to disappoint you, because the eePASSIGE system still presents some small limitations.
In fact, inserting cargos larger than 10 kb still results in low efficiency, and also the locus and the cell type can negatively affect the efficiency of the system.
Nevertheless, this new technology is so far as good as it gets with integrating large DNA sequences. Hopefully, further evolving the recombinase could deal with these issues as well. When they do it, we here at CRISPR will be ready to tell you all about it.